Hi everyone, this is Nicole Freeman, and this is going to be a summary of kidney function at the nephron. So remember, the nephron is a tiny microscopic tube that you have like a million or more of in every kidney, and that's what's actually filtering the blood and then making urine as a result. And so we're going to draw some processes that occur along the tube. So the first thing that we need to do is to draw a kidney nephron. So here goes. I'm going to try to make this not look too crazy of a picture. So you can follow along with me if you all would like, if you have a blank piece of paper too. So then it's going to go down. Okay. This is going to be the loop of Henle. This is going to be the capsule, the PCT, loop of Henle, and then we're making the DCT. This is not looking awesome so far, but that's okay. All right, it's kind of hard. I'm trying to get around my little tripod here. And then all the nephrons hook up to a collecting duct, so we'll go ahead and show that. So this is going to be the collecting duct. And the collecting ducts, remember, there's not just one collecting duct per nephron. There's tons of nephrons that can hook up to collecting ducts. So that's kind of, we'll just sort of show a branch like that and another branch like that, meaning that more nephrons could be hooked up all over the place. So let's go ahead and sort of draw. This is the outer layer of the capsule. That would be the parietal layer. And this would be the inner layer of the capsule, which I can't draw it like this, but this would be directly touching the glomerular capillary bed that goes right there. And so that's actually what we'll draw next. Okay, so here is an arteriole. It's going to be our afferent arteriole, which leads into the capillary bed, the glomerulus. And then remember that you don't have a venule coming off of the glomerulus. You, like most capillary beds, you have coming off of it a venule. We don't in this case. It's just another arteriole. Okay, so let's go ahead and label some of these things. I have a little bit more I want to mention about where this goes in a second. But here, this is the renal corpuscle. Down here, I'm just going to kind of write the labels up above. This is the renal corpuscle up here, which is just made up of those things: capsule, glomerulus. Then right here is the PCT. And then right here is the loop of Henle. I can write that down here, I guess. Or nephron loop. As long as you have a loop in the name, that's all that matters. Loop of Henle. This is the descending limb, and then this is the ascending limb. And then we have the DCT over here. And then we have the collecting duct right here. Collecting duct I'm putting in parentheses because it's not really part of the nephron. The nephron stops right here. So let me just make sure that you guys remember some basics. So I have this nephron, which is this tiny tube that you have a million of in the kidney. I have it drawn out, very kind of almost spread out, so you can see each part really easily because we're going to draw a bunch of stuff on here. Is this really the way that it looks in the kidney? All these nephrons, are they sort of like from left to right, corpuscle, PCT, loop of Henle, DCT? No, it's usually like much more twisted around and things are going up and down and all that. But for simplicity's sake, we're leaving it like this. Okay, let's go ahead and again label some of our other things. This is efferent arteriole. This one's the afferent arteriole, which is leading blood into the glomerular capillary bed. Efferent arteriole drains the glomerular capillary bed. Now, this is what I wanted you guys to remember, is that I'm not going to draw what this continues on to be, because you know why? It's going to make my picture look crazy, and we'd have to do a lot of writing on here today. So what is the efferent with the E? What does the efferent arteriole lead into? Another capillary bed, right? So this one right there is the glomerulus, which is a fenestrated capillary bed that's there for what? Step number 1, glomerular filtration, right? You're going to push stuff in from this capillary bed into the nephron. We would call it filtrate then, and then you're going to go through and do all some fancy stuff with the fluid that enters in here. But what do we have on the other side of this? Another capillary bed called the peritubular capillaries. right? And so where do those go? What does peritubular mean? Around the tubules, which this is the tubule. Remember, proximal convoluted tubule, distal convoluted tubule. So this keeps on going, but I'm not going to draw it like this. This keeps on going, and this just covers all of this stuff. That's really, really important that you remember that even though we're not drawing it, there is a very close relationship with the blood, which is like right here, flowing by in tubes, and the filtrate in the nephron right here. Okay, so just remember that that continues on everywhere, but I need to do writing, so we won't draw it. Okay, so let's go ahead and start with step 1. So what we want to first consider is what can we possibly find in the filtrate that can be pushed from the blood in the glomerulus into this tube right here. So we're going to write filtrate under here. What is in the filtrate? Well, what do you guys think? I would hope that as we're drawing this video, writing this together and talking on this video, that you would be thinking of some things. The key is what? Whatever we're going to have in here, it's only going to be things that are what? Small. Okay, this process here at filtration that happens in this step 1 to making urine, it's not selective based on like, what are bad things that we need to take from the blood and put them in here, or whatever. It's not like that. It's just size selective. So you're going to have anything small enough that can squeeze through the tiny openings in the capillary wall, those fenestrations. So first of all, water is going to be small enough, and we should definitely consider that because you're going to have more water than really anything. That's the main component of blood plasma, right? So it's going to be easy to push out into the nephron. So then you're also going to have some nutrients. Like, is glucose small enough to get pushed through the glomerulus into here? Sure, it's small enough, so we'll have glucose. What about amino acids? Yeah. Okay, those are our nutrients. We can kind of write that because it'll be easier just to say nutrients later, NUT for nutrients. What about ions? Sure, you're going to have ions. So first of all, salt is going to be in here, NaCl, and remember those can break apart into Na plus and Cl minus. You're going to have some other ions, like what else? What other kind of ions would you have in the blood? You'll have, let me squeeze my hand in here like that. There we go. This is easier. Bicarbonate ions are going to be in the blood. Hydrogen ions are going to be in the blood. Calcium ions are going to be in the blood. Potassium ions. Okay, you're going to be some drugs are going to be in here, like drugs like medications maybe that you might be taking. We might have some toxins that, I don't know, just random toxins that we create, or if we want to be really specific, we could name some wastes. Like what kind of wastes do we have that we talked about? Like nitrogenous wastes, for example, would be like ammonia, ammonium, urea, uric acid, and then like creatinine would be another waste that we get from muscle metabolism, stuff like that. So all these things, they're all small enough to get pushed through the capillary wall of the glomerulus into this tube. So all of this is going to be passing through. Now this is kind of a problem though, because while there are some things on this list that are wastes and toxins and stuff that we don't want, there are also a whole lot of stuff on here that are good. So that's what the job is here for this tube, is to make sure that we, like some of this good stuff, we actually reabsorb, which means take it from the nephron and put it back into the blood, specifically into the peritubular capillaries that are going to be passing all through, passing all along right beside this tube. Okay, does that make sense? I hope it does. So let's go ahead and start with that. Step 1 was filtration, which was pushing stuff from the blood into the nephron. Now the second step is going to be reabsorption. So I'm going to draw reabsorption occurring in blue. And so with all the things that we talk about that are reabsorbed, remember those are going to be the good things that you don't want to like end up keeping in this tube the whole way, because then if you do, that means it's going to be urine in the end. We should actually write that. Whatever we get down here, this, oops, you guys probably can't see that. That's going to be urine. So you don't want to leave all the good stuff in there to the end, because then you're going to pee it out. So we're going to draw our arrows going out like this. So we're going to draw a little key down here. This is going to be in blue. I don't know if you guys can tell that this is blue. This is a blue pen. I don't know. Does it look any different than my than my black pen. I don't know. Maybe I'll highlight it or something in a different color later. Like I'll post the picture or something on Blackboard. But anyway, so the arrows going out mean that that's what's being reabsorbed. Remember, that's going to be the good things that we want to put where? Boop, into the peritubular capillary, so that we give it back to the blood, back to the body. So this is all the things that we're going to draw in here that are reabsorbed. This is all summarized in a slide in your lecture notes, but it's just nice to be able to imagine where it's happening. That's the purpose of me doing this video. So first of all, almost all nutrients are going to be reabsorbed and given back to the blood right here, right away. This is good. We don't waste any time to take those back. And our nutrients are things like glucose and amino acids. Do you want to keep glucose and amino acids in this tube the whole time and then pee it out in your urine? No, these are the building blocks for the organic molecules that keep you alive and make you who you are, make you be human and do all your bodily processes and everything. So you want to keep those. Okay, so we're going to give those right back to the blood. Almost virtually all the nutrients are going to go back into the blood right there. Then we're also going to do a lot of water. But the reason, one of the reasons why water is so successful to be reabsorbed right here at the PCT is because we reclaim about 65% or reabsorb about 65% of sodium. Okay, so that's great, because then what that's going to do is allow water to follow, and then you take back a bunch of water right away. What else is going to follow sodium besides water? Remember, water always follows solute, right there. Okay, what else is going to want to follow water is, follow sodium, is this little guy, the chloride ion, because it's negative, it's positive, they're attracted to each other. And then depending on the needs of the body, this could be a place where you might reabsorb some bicarbonate ions, the HCO3 negative. The reason why you would reabsorb some bicarbonate ions here would be if your blood pH was getting too low, because this acts as a base, which can make it be brought back up a little bit. So this is just if you need to, okay? So now let's look at the loop of Henle. So that's a lot of things, like out of this list right here. When you look at the things that we could have in the filtrate, we start taking back a lot of it right away. And in fact, you guys should make a mental note that most reabsorption happens here at the PCT right away. Most reabsorption happens right here at the PCT. But we don't stop reabsorbing all along the way. We continue to do it, and even at the collecting duct too. Okay, so let's talk about the loop of Henle. So what happens is, as you're going down on the descending limb of the loop of Henle, you have it permeable to only water. So let's draw our arrows out again. Remember, these peritubular capillaries up here, they're going to go all around here. Might even dip down a little bit and then come back up. And so when you show this arrow moving out, that means that's going to go where? In the blood. Okay, and oh, I should mention something about water. We need to mention that up here too. So the water reabsorption, both at the PCT and at the descending limb of the loop of Henle, the water reabsorption is accommodated via aquaporins. Aquaporins are these tiny little water channels that get inserted in the membrane right here, you know, all around, so that it makes water have a place to travel through really, really quickly. All of the cells making up these areas, I mean, a cell membrane is very lipidy, so water wouldn't travel through there as quickly as it would in aquaporin. So water reabsorption is very efficient here at the PCT and here at the descending limb of the loop of Henle because of aquaporins. In fact, what you want to call this up here is obligatory water reabsorption, because of the presence of the permanent, you have a permanent residence, a permanent insertion of aquaporins all the way around here. Okay, so again, I know I kind of went back to describe something at the PCT. We're really at the loop of Henle right now. We made it all the way down to here. And so remember that the descending limb is permeable only to water, no solutes. That actually happens on the other side. And there's a reason for that. You don't ever want there to be, you never want the solutes on either side of the membrane to like equal out, because then you wouldn't get any movement in either direction. This is all based on diffusion and stuff. So you got to make one side designated just for water, and then the other side designated just for the solutes. So this is permeable only to the solutes, which are primarily ions that we're talking about here, like sodium, maybe some potassium, if you need some, chloride, even magnesium. I don't know if that was on our slides before, etc. Okay, but no H2O. Okay, water is not able to pass through the ascending limb on the other side. So water is designated for this side. The solutes is this side is designated for the solutes on the other side. Okay, so so we're making our way through. Okay, now we're at the DCT. And so remember, the filtrate, it keeps on going, right? It's passing through these tubes, and all the while we've lost some stuff, which is not bad because these are good things, right? These are things that your body needs. But we're not going to stop doing that here. Whatever has reached this point now, we still want to pull from it and take some things back into the blood. So let's draw some familiar things. So we have, are we going to do water again? Oh yeah, water is going to be reabsorbed all places, all 4 places, PCT, loop of Henle, DCT, and even the collecting duct. So water for sure, but I want to make sure that everyone knows that this DCT, we have to remember the hormones because this is hormonally regulated, the reabsorption and secretion here. We didn't need to rely on hormones for the reabsorption of materials so far because the concentrations of them in the solute are so high, they just will easily diffuse into the blood where they are in lesser concentration. But now we need a little help. We need hormonal action to continue to make sure that we are reabsorbing enough. So water reabsorption, this is what hormone is going to help us with this? ADH. Okay, and so remember over here, did we need to put in ADH to make water reabsorption happen at the PCT? No, because aquaporins are always present here. So this is called obligatory water reabsorption. But over here, now this is called facultative water reabsorption because only when ADH comes into play do we insert the proper number of aquaporins so that water can move across into the blood efficiently. All right, so how, besides the presence of the aquaporins via the ADH, what else is going to facilitate nice water movement, water reabsorption into the blood? Of course, the sodium reabsorption. And so you're going to reabsorb sodium along with the water just like you do every other place. That's what's going to make water follow. But here we need a hormone for this guy too. Aldosterone is actually going to make water reabsorption happen more at the DCT. ANP, called atrial natriuretic peptide, ANP, we can actually circle it and sort of like cross it out because that just to remind us that this whole thing would shut down if ANP came into play, atrial natriuretic peptide. It's just the opposite acting hormone to aldosterone. All right, and so how else is going to be reabsorbed here? Chloride, again, because it's attracted to the sodium. Okay, and we're forgetting another one too, another important one, which is calcium. Okay, and what hormone? Again, this is all hormonally regulated. PTH. PTH secreted from the parathyroid. This is going to target many places in the body, but one of them is here. This is going to raise blood calcium levels by making sure that we don't urinate out the calcium that's been put in the filtrate here in this tube, but rather put it in the blood. Okay, and so what you see happening at the DCT, I guess besides calcium, this is really the only place where you see the calcium reabsorption, but all of the others, like ADH and aldosterone, causing water and sodium reabsorption to happen here, you also see that happening on the collecting duct too. So let's go ahead and we can kind of draw those again. So sodium, this is via aldosterone. Isn't that great that we actually have, we continue on? I mean, look, whatever has made its way through the whole tube from start all the way down here to finish, we're almost done. Like whatever makes its way to the end of this tube here, the collecting duct, it's urine, so it's almost done. So we still like do the last chance, sort of last ditched effort to get some good things taken from it and added to the blood before it becomes urine. And so of course what's going to follow the sodium is going to be water and ADH. This is also be the facultative, just like we see with the DCT, facultative water reabsorption. And then of course we're going to get the chloride ion following the salt, and then I'm actually going to draw an arrow out like this because maybe some hydrogen ions or bicarbonate ions, it just depends on pH. Okay, and so actually when we use our other pen to do secretion, which remember is going the opposite direction, taking things from the blood and adding them to the urine, adding them to the filtrate, which the arrows will all go like that in that case, you can see that I can sort of make the arrows, oh, sorry, I just looked at the video. There, can you see it better now? Okay, we can reabsorb some hydrogen ions or bicarbonate ions here into the blood, just depending on the pH, but we could also secrete them here too. That's why I have the other arrow going in the other direction. This is indicating you can both secrete or reabsorb hydrogen ions and or bicarbonate ions at this location, but I can't tell you which one you're doing right now because it just depends on what the pH is of your blood and bodily fluids. Okay, so now that's kind of getting us into this next talk, which is going to be secretion. Okay, so secretion is just the opposite of reabsorption, where we're going to be taking from the blood and adding to the nephron filtrate. So remember, like I said before, what we're missing from this picture is these peritubular capillaries going like this and traveling all around the tube, you know, like that. And so when we show these arrows like this, I'm going to use my black pen for the secretion. Okay, we'll just kind of go like this. Again, I don't know if it really looks like a different color to you guys. Maybe I'll highlight it in yellow or something in a second. But when we draw our arrows like that, that means that these things are being secreted, which means it's the bad stuff that we kept in the blood, didn't get added to the filtrate here at step 1 during filtration, but it's still in the blood. And so now we need to like add it in now. Okay, so what are we going to happen? What's going to happen at the PCT is going to be a lot of secretion. This is where most secretion occurs, just like how this is where most reabsorption occurs. Whoops, something happened to my computer. Okay, it's still working. All right, so this is where you're going to have like some drugs be added to the filtrate from the blood. Maybe it's not like bad drugs, just like medications, but your body thinks it's bad, so you just put it in. Okay, then this could be where like a nitrogenous waste like ammonium ion could go in, or maybe some hydrogen ions because maybe you have too many, or maybe some toxins. So there's going to be a whole lot of stuff added to the mix right there. Okay, a lot of urea can be secreted here at the ascending limb of the loop of Henle, and then at the DCT you'll have just waste in general. Just it could be a whole slew of things, creatinine, maybe more urea, something like that. But something else that I want to put here is secretion of potassium. And so you really, in order for your cells to be working properly, you need to have a perfect balance of sodium ions and potassium ions distributed in and around your plasma membranes. And so if you have too much potassium, that can be dangerous. So you're going to secrete excess potassium. And actually, you're going to do this in both places, okay, both the DCT and the collecting duct. So whatever you learn for the DCT, it's very similar for the collecting duct too, with the, of course there's a couple of differences, like looking at the calcium and stuff. But anyway, yeah, whatever typically happens at the DCT, you're going to have happen at the collecting duct. And this is also underneath hormonal regulation. So which one of these hormones, ADH, aldosterone, ANP, or PTH, do you think is responsible for aiding efficient secretion of potassium at these two places? Pick one of these. Which one? It is aldosterone, which we talked about in the renin-angiotensin-aldosterone mechanism video. We usually talk about aldosterone, when we mention it, we usually talk about its effect on sodium reabsorption and water reabsorption then, and then therefore its effect on blood pressure, but it also aids the secretion of excess potassium. So there's a whole lot there. Okay, and then to finish secretion, you might secrete some ammonium ions right there at the collecting duct. And then just remember that you could, like we mentioned before, you could be secreting, not reabsorbing, like we just finished talking about, but you could be secreting hydrogen ions or bicarbonate ions and adding it to the filtrate depending on the needs of the body. So here's an example. of how this could kind of like go back and forth here, where you could be reabsorbing 1 minute and then secreting the next or whatever. So let's just say like if blood pH is dropping. Okay, if blood pH drops, what does that mean? Blood pH is getting, it's like we're going down from like 7 to the 7.45 to then like 7.4, then to like 7.3. That would mean it's dropping, right? Which means it's getting what? More acidic. Okay, and remember, acidity is just hydrogen ions. So if blood pH is dropping, then what we would want to do is the kidneys would secrete the hydrogen ions, right? Because that's what's making the blood acidic and making the pH drop. So secreting it would mean getting it out of the blood and adding it to the urine so that it can't make the blood acidic anymore. And then on the same token, as it's secreting the hydrogen ions to get rid of the H+, you could also reabsorb at the same location the bicarbonate and put that in the blood, because what that's going to do then is a bunch of bicarbonate floating around will pick up any leftover hydrogen ions in the blood and drive the pH down, drive the pH back up, make it a little more basic. So anyway, this is just an example. If blood pH was dropping and becoming too acidic, then the kidneys would secrete hydrogen ions and reabsorb bicarbonate. If blood pH was rising, which means it was becoming too basic, then just the opposite would occur. Okay, and so that is the summary of the nephron function of making urine. And so by the time you get to the urine, now we're ready to have it get out of the kidney and then get down into the ureter and be stored in the bladder until we release it through the urethra into the toilet or ground if you're going to the bathroom outside. So by the time that this is all said and done, the urine is about 95% water and about 5% solutes, could be urea ions and things like that. So anyway, I really like drawing it in a picture form, and I'll take a picture of it and post it on Blackboard so that maybe you can like look. I kind of wrote small in some of these areas, so maybe it's not really legible yet. I'll post a picture so maybe you can see it better. I think it just really helps for studying purposes. This is what a lot of the questions I could pull from here. This is what I want you to know, all these little notes that we've made here. So anyway, I hope that this video helped just summarize things for you, and I hope that you're understanding it a little bit better now on how the how the kidneys make urine. So you guys have a great day, and contact me with any questions as always.